So they will look at the enzyme inhibitors. Now, what are basically enzyme inhibitors? Basically, inhibitors are the substances that retard the rate of reaction. Actually, they slow down the rate of reaction. Substances which retard the chemical reactions are known as enzyme inhibitors, right? So there are three types of inhibitors. The first is competitive inhibitor. What happens in this basically? Okay, it's wrong spelling. Um, it doesn't come here. Competitive inhibitors. Now, what happens in this? Basically, in this, okay, here the inhibitor you see in the given diagram. Here, this is a substrate, and this is the active site, and this is a normal substrate. This is normal binding of substrate, right? This is the normal binding of substrate. Now, what happens in this inhibitor which has the same size and shape and charge as the active site resembles to the substrate. It resembles to the substrate, right? For example, this is the substrate, and it attaches in the side of that substance or that substrate. Basically, what happens? The size of inhibitor is similar to substrate, right? So it actually to substrate basically, right, is similar to substrate. So what happens? It attaches with the active site and it blocks the active site. It blocks the active site, right? As you can see over here, it has attached with the active site, so it's quite unable for the substrate to attach with it. So that's why they are known as competitive inhibitor. They actually block the active site. Okay, they are actually reversible. They are reversible. What does that mean? It means that after some time this inhibitor will deattach. But what will do? It will actually the increase. It will decrease the rate of reaction. But after some time it will get managed, right? So now basically when we talk about graph, now let's talk about the graph, right? Okay, as you can see over here in the given graph. Okay, okay. As if you can see, this is the graph that when we was increasing the substrate concentrations, you have seen in the my previous video about the substrate concentration and the factor affecting enzymatic activity. So as you are increasing substrate, the enzymatic activity is increasing. But what happens when we are adding competitive inhibitors? First of all, in substrate, obviously the rate of reaction will decrease, right? But after some time it will increase as we are increasing the substrate concentration. Okay, actually this depends upon number. This depends upon numbers. Now how? For example, if we are having two inhibitors, right? They are having two inhibitors, and this is a enzyme basically, right? This is the enzyme, right? And we are having one substrate. We are having one substrate. So it is obvious that one inhibitor will come and join the enzyme. But what if we increase the substrate concentration and we make it to 5? we make it to 5. So obviously it's obvious that now this substrate will overcome this inhibitor, and this substrate will join the active site. So this happens when we increase the substrate concentration. The enzyme reaction will also increase, but after some time, slowly and gradually. Now, it is at this point there is Vmax. It means velocity is maximum. Now you'll be wondering that where is this? Now you'll be wondering that what is this V half upon 2? Actually, V upon 2, which means that when the velocity of the Vmax upon it is actually Vmax upon 2. When we divide the Vmax upon 2, for example, we have Vmax is 5. Like, okay, and when we divide Vmax upon 2, we get 2.5, right? So now what is this km? What is this km? Basically a point at which that there is Vmax by 2, at which a point at there is Vmax by 2, that point is known as km. That point is known as km, right? And it is not included in our post. It was just first because it was included in the graph. So I hope you understood this graph. Okay, for example of this is basically we are having one enzyme which is known as succinate dehydrogenase. This is basically an enzyme which basically converts succinate into fumarate. That converts that converts that converts succinate into fumarate, right? It converts that enzyme into fumarate. But over here, the competitive inhibitor, which is the competitive inhibitor, the competitive inhibitor over here is malonate, which comes in between the reaction, and it retards the rate of reaction. So basically here the malonate is competitive inhibitor, right? This malonate is competitive inhibitor, right? And one more important thing. Okay, another example of this is sulfonamides. Sulfonamides. Basically, these are some antibacterial, these are actually drugs, right? Which are used as which are used as competitive inhibitors. We are also having penicillin. This is actually a antibacterial which is used as competitive inhibitor, right? Now next we are having is competitive. The number two we are having is, okay, basically before moving ahead, you must know that this competitive inhibitor follows lock and key model. It follows lock and key model. And one more example of them is feedback inhibition. Sorry, over here it would come. That is not found. So basically this model, basically this competitive inhibitors follows lock and key model, right? Now let's move to number second, which is non-competitive inhibitors, right? We're having non-competitive inhibitors. Now what are they? Actually, the substrate attaches to the allosteric site of the enzyme, which changes the size, or you can say which changes the shape of active site, enabling them, like the enzymes are unable to enzymes are unable to get attached, right? Okay, now what happens basically is, consider that there is an enzyme, and okay, this is the enzyme basically, right? And here a substrate is there. There is a competitive inhibitor and attaching to this side. Now this is not active site. This side is known as allosteric site. This side is known as allosteric site. A site other than the active site is known as allosteric site. So when we are having the substrate whose size is like this, the size of like this, right? So it will be easily attached, right? Like this, complementary to the shape or to the size of enzyme. But when this non-competitive inhibitor joined, what happened? It changed the size of basically it changed the size of the enzyme. Now what happened? This enzyme has become now like this. Now this can't join, right? So what happened by the joining of this non-competitive inhibitor? It changed the shape of the active site. Enzymes are able to get attached. Now this is the non-competitive inhibitors basically, right? Now if we talk about the graph of this is, we draw the graph. Okay, on normal reaction on increasing the substrate, it increases, right? But when we talk about this, when we increase the substrate, this will never reach the V max. It will never reach the V max. So they are known as irreversible. They are known as irreversible inhibitors because they can't be reversed. Basically, non-competitive inhibitors can be reversed and cannot be reversed. Basically, if we talk about feedback inhibition, so feedback inhibition is reversible, 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 non-competitive inhibitor. An example of non-competitive inhibitor, right? So, for example, we have some examples too. Um, for example, we are having cyanides. Cyanides. Basically, these cyanides combine with actually with cytochrome oxidase cytochromes. In actually, they retard the cellular respiration. Next, we are having is heavy metals. Heavy metals are, for example, Hg plus 2. For example, we are having other heavy metals. For example, okay, there are many. For example, manganese, copper. We are having copper. So basically, what they do, these heavy metals combine with thiol group, with thiol group, which is basically SH group. It combines with them and it breaks the it breaks wait actually it breaks disulfide bonding between tertiary structure. Now, what is this? Basically, we know that the enzymes are made up of tertiary structures, and tertiary structures are supported by disulfide bonding. So, what happens? These heavy metals combine with thiol group, and they try to break the disulfide bonding between the tertiary structure. So, resulting the destroy of the enzyme, right? Now, next we are having another type of which is known as uncompetitive inhibitor, which is known as uncompetitive inhibitor. What happens in this, as we know that, for example, this is a substrate, right? This is the enzyme. Now, this is known as substrate, this is known as enzyme, right? And when they both combine together, they form enzyme substrate complex. They form enzyme substrate complex. So what happens basically between this enzyme substrate complex between an enzyme substrate complex and inhibitor comes in here, right? So this type of inhibition is known as uncompetitive inhibitors. Basically this type of inhibitor is only given in the synth book, in the new synth book. It's not given any other. So I hope this is clear to you. If not, you can comment down. I will be very glad to answer you guys.